What's up, Will? Hi. Today we're going to be talking about DNA technology. So when we talk about DNA technology, we're talking about the technology that we have related to the use of DNA, genes, genetics, genetic modification, and those types of things. So let's go ahead and start right into our essential questions. Number one, what are the two historical biotechnology tools that were used and explain how they're opposites? Number two, what is a cell transformation? Now, we have an ed puzzle that will help go through some of this as well. Number three, what is recombinant DNA? Okay, the name kind of gives away a lot of that, and this will also be included in that ed puzzle as well. Number four, what are transgenic organisms? And is this inter or intra-species genetic engineering? And lastly, what is an example of a transgenic bacteria, plant, and animal? Now, before we start this, hopefully you've heard of GMOs before, these genetically modified organisms. We've been dealing with them far more often than you probably realize. We've been eating them as part of our diet, everything from the fruits and vegetables that we consume to the meat that we eat to the dairy that we drink. Um, but in addition to GMOs in our diet, DNA technologies affect our lives in many different ways. We use gene cloning and editing to produce medical industrial products, everything from vaccines and human byproducts to go ahead and use for those that are deficient in manufacturing them themselves, to oil eating bacteria. All right. We use DNA profiling, which has changed the field of forensic science, forever changing law and legal investigations. And lastly, bioinformatics has provided huge amounts of research data to allow for historical and evolutionary investigations over time, looking at allele frequencies and the fluctuation of patterns within species. Today, we will try to highlight just a couple of these applications and techniques. This is not exhaustive because there's a lot that's going on right now, but we want to hit some of the key concepts that have been big historically, start looking at what the future might entail, and kind of give you some of the key vocabulary to make sure you understand the concept of genetic engineering. One of the key terms that was really big when I was getting done to graduate college in the year 2000 was this idea of biotechnology. These big biotech firms were taking off all over the place and they were flooding, especially places like Silicon Valley down in California. And while it seems like a very modern field because of that boom around the year 2000, the manipulation of organisms and their components to make useful products, which is what biotechnology is, actually dates back to the dawn of civilization. If we start looking at ancient practices such as the use of yeast to make beer and bread, I mean, let's think about this. Beer, the first evidence we have of brewing beer actually dates back to about 3,900 years ago when we were looking at a poem honoring Ninkasi. Yes, the same Ninkasi that the beer is named after, who was the patron goddess of brewing. And it contains the oldest surviving beer recipe. We know that Egypt, they were doing first leavened bread with yeast, and this dates back to around 1000 BC. So these are pretty old practices. Grafting fruit trees. The first orchards to use some sort of root division existed around 4000, 3000 BC, and somewhere between there and 1000 BC, humans developed the ability to do fruit tree grafting. The exact date's largely unknown, but this is an advance that largely reshaped the world of cultivation, as you could get identical clones of the actual plants that you liked. So if you liked a taste of a fruit, you could go ahead and make more of that fruit. And that's not even including the selective breeding of livestock, dogs, or other animals, which we can see in many different facets on our planet. So let's go ahead and start out with our historic modes of natural select of biotechnology. So The first is what we call selective breeding, and this is when humans kind of choose which organisms breed to produce the desired traits for the next generation. Humans use selective breeding to get what they want from the organisms that they have. And good examples of this are our purebred dogs. We were talking about the term pedigree before. We can start looking at the ancestry to most of the dog breeds being pretty closely related. And then they diverge as humans start to go ahead and tamper with the breeding stock to go ahead and develop the gene pool that they want for the desired traits. We start looking at the same thing for plants. You start thinking about greenhouses as a place where we're going to go ahead and control the genotypes that end up producing the future offspring. So selective breeding can be used to reinforce traits or can be used to create variation. And there's a great documentary from Nova called Dogs and More Dogs that looks into the genetic diversity of canines, our friends, and creates, we look at all the variation created by selective breeding. So if you're interested in this type of material, I'd encourage you to go ahead and check out that episode of Nova. Now, the two most common methods of selective breeding are hybridization and inbreeding. And you've probably heard both of these terms. A hybrid is something that's a cross between two. So hybridization is the process of creating this mix. And then inbreeding is exactly what it sounds like. You're taking species, individuals that are very closely related to each other and breeding them together to try and maintain the same results that you've been getting. Hybridization is crossing dissimilar individuals, and the offspring, called hybrids, often produce a new line of organisms with those desired traits. You see all sorts of hybrid plants and flowers. In fact, if you go to Jerry's or Lowe's or Home Depot to get your plants, probably most of those are hybrids. Okay. You also see here in the picture on the bottom right, labradoodles, but there are tons of other ones. We can start looking at any times of crossbreeds, and you get these new hybrids, some of which become their own kind of popular breeds later on down the road. So in these cases, we're taking things that are not alike, breeding them together to get new combinations that didn't previously exist. The opposite of what we see with inbreeding. Okay. So inbreeding is continued breeding of individuals with very similar characteristics. The many different breeds of dogs are a good example of inbreeding. That's how you get those consistent traits over time. And although inbreeding does retain the desired characteristics, there are risks associated with it because you're making the gene pool smaller and smaller over time. This limited variety of options can lead to the increase in recessive genetic defects. So let's take a look at these two organisms. First, you see on the top the Dalmatian. Now, does anybody ever wonder why the Dalmatian has been associated with fire departments? They were actually selected for this job because it used to be that before there were sirens and engines, they used to have a dog run around in front of the truck and bark to let people know that the fire truck was coming. Once they developed sirens, they had no need for the dog anymore, but a lot of the people that worked at the fire department still liked having a dog. Problem was is the big sirens scared a lot of the dogs off. Well, it turns out that there's a fraction of your Dalmatians that actually are deaf. They can't hear them, and this is why they were selected by the fire department as their mascot. They still got to keep a dog around, but they didn't have to worry about the dog being afraid of the sound of the sirens. The picture below is the family known as the Fugates. You may also have heard of them as the Blue People of Kentucky. This was a family that lived in the hills of Kentucky and are known to have been carriers of a genetic trait that led to the blood disorder, methemoglobinemia. I don't think I said that right, which causes the appearance of blue tinged skin. And so their bodies actually produce this slight blue tint to their skin. Again, these are genetic recessives, but because the gene pool gets too small, the frequency increases. Now, when you think of genetic engineering, when you think of biotechnology, you're probably not thinking of hybridization or inbreeding. But these are historic examples of these two technologies. Okay. When people use the term biotech today, they are usually referring to direct DNA technology, modern lab techniques for studying, manipulating genetic material. This has been an ongoing process ever since Gregor Mendel's original experiments in the inheritance of pea plants in 1857. The theory of genes controlling our physical being first came to light. This evidence gave firm affirmation to the as yet undefined gene carries hereditary information from generation to generation. All right. Now, when genes then are responsible for the genotype and through proteins, as we've learned earlier in this semester and last term, ultimately the phenotype of the individuals. So this transfer from genotype to phenotype occurs because the instructions are found in the DNA and those are converted into proteins, which give us the physical characteristic. 4 decades later, in 1902, Walter Sutton and Theodore Boveri proposed that chromosomes, the superset of DNA, were responsible for carrying the genetic information. That would later be built on in 1953 by Watson and Crick, giving us the double helix structure that we talked about. Again, only possible because of Rosalind Franklin's X-ray crystallography picture. A series of four-lettered bases then, A, T, C, and G, make up the nucleotides that are going to go ahead and give rise to our potential traits. So this brings us to all that we kind of know already, that we have this structure of DNA, where in humans at least we have 46 chromosomes, two sets of 23. That is the diploid number. And that if we were to stretch all of that DNA out end to end, we'd be looking at about 6 feet in a particular cell. Those genes are made up of nucleotides that get expressed in the real world by converting the nucleotide base pairs into amino acids and ultimately into the folding of the protein. Now, we could go ahead and spend a lot of time going through this history of genetics. I'll go ahead and leave a copy of this for you on our website. But essentially, we get out back to the 1900s where people rediscover Mendel's laws, which eventually leads to the development of genetics. 1953, Watson and Crick identify the double helix as the chemical basis of heredity. 72, DNA recombination first starts to occur. This is a big one because this is when we start making our first transgenic organisms that we'll talk about here in a second. 1980, we have DNA markers so that we can start mapping the human genome to identify particular disease genes or at least chromosomal regions. Then we start looking in 1983, we identify Huntington's disease. 1990, we begin the Human Genome Project. And that represents a huge milestone that kind of ushered in a huge plan to map the whole sequence. Prior to this, we'd been working in small labs that allowed us to go ahead and focus on small little sections of maybe a single chromosome. But the Human Genome Project ushered in cooperation that reduced the costs and allowed access to information to, people all over the world. As this process is ongoing, you start seeing the genetic and physical mapping process occur. 94, 98, 98 DNA markers are used to map human diseases to particular chromosomes. By the year 2000, we had our first working draft of the human genome sequence. In 2000, we also discovered the CRISPR process, which we'll talk about later today and tomorrow, which becomes a huge revolution in what we can do to edit that DNA. In 2003, the Human Genome Project announces that it has a complete genome map. And in 2005, they discover the other half of the CRISPR-Cas9 technique by discovery of the Cas9 and PAM, which is a huge advance. And when we combine CRISPR and Cas9 together in 2013, we're able to harness this for genome editing. So we've had this technology for about 7 years, although there are limitations in what we allow to happen and how successful we've been so far with that type of change. that's kind of an overview of the process that kind of leads us to where we are now. So, this is genetic engineering in a nutshell. So, with all this information that we now have, researchers have realized that it's possible to develop technology for the addition, change, or deletion of specific genes, um, which can then quite simply and naturally lead to the development of a new science. Okay, that's a big deal. That's no joke. Genetic engineering mainly requires a gene to be transferred into a host cell. Historically, we used a vector of biomaterial um, in terms of like a plasmid or something, and we'll talk more about that in the Edpuzzle video as well. But that's changing. This idea of transforming a cell by putting foreign DNA into it, a cell transformation, is becoming more and more common and cheaper and easier in many respects. So, a good term to remember here is what we call recombinant DNA. This simply means DNA that's been recombined with the original DNA and whatever new DNA has been inserted into it to hopefully create a better organism. Now, I've got a couple little videos at the end of this for you to watch. So uh, I'll go ahead and play the first one here, talking about this process of designing genes.
Scientists first learned how to change genes in the early 1970s. Using the techniques we're about to describe, it's now possible to take a gene from a lizard and mix it with the DNA of a plant. We can take the genes of jellyfish and mix them with genes from mice. This is recombinant DNA technology, and it works because the genetic code in all living things is written in the same way. It's like a universal language. We've mentioned that people suffering diabetes use human insulin made by genetically manipulated bacteria. Rennet, an enzyme originally taken from calves and used in cheese making, is made in a similar way. Both insulin and rennet have been relatively easy to produce using genetically engineered bacterium because bacterial cells contain isolated rings of DNA. We call these plasmids, and they have some special properties. First, plasmids are taken from bacterial cells and mixed with a restriction enzyme. These tiny proteins act like molecular scissors, able to recognize and cut a specific base sequence. When this happens, 3 bases are left unmatched at either end of the severed gene. These are called sticky ends. In the same way, the gene to be joined to the plasmid is isolated. Now the gene and the broken plasmid can be joined together by mixing the broken plasmid and isolated gene with another enzyme called ligase. Ligase acts like glue, and it causes isolated DNA strands to join at the sticky ends. The altered plasmid is then reinserted into a bacterial cell. And as the bacterium divides, the modified DNA is copied exactly. Most importantly, the bacterial cell machinery will now make a new protein, as defined by the introduced gene.
Okay, so this is the basics of what we call recombinant DNA technology. And our Edpuzzle will talk more about this in a little bit more detail. But essentially, this leads to one of the key differences in terms of how we use genetic engineering. From a practical application, um, this could be good in a lot of different fields, from crops to livestock to biologically based consumer goods, things like cotton. There are two basic varieties that we start looking at. One is what we call intraspecies. And this is within the same species. So taking like one human gene and putting it into a different human, but still within humans. And then we have interspecies, where there are two or even more species, where we insert the gene from, let's say, a jellyfish into a mouse, as they mentioned in the video. This would be called interspecies because we're transferring DNA from one organism into another organism. So again, intraspecies, this is within a single species creating recombinant DNA to produce literally next generation results that might normally take decades or even centuries to occur in traditional plant and animal breeding, but we're able to do it in a much faster time versus interspecies, which is going to be improving a species by inserting a gene or genetic characteristic from one organism into another organism. A good example of this is the use of what we call Bt. It's a type of bacteria, Thuringiensis, which is used as a pesticide in cotton and potatoes. They had Bt cotton, Bt potatoes. The cotton is still available. You've probably even worn some, but we don't make the Bt potatoes anymore because there were a lot of people complaining, and companies like McDonald's wouldn't take it anymore. Now, if you're interested in that type of thing, there's a great episode of a mini series called The Botany of Desire with Michael Pollan that goes into this kind of genetic engineering. So look for the episode on the potato, and it'll talk about human in investing and looking at genetic engineering, a much different relationship with the plant to what we'd seen before in terms of selective breeding. Now, whether you're doing intraspecies or interspecies, the ultimate product is what we call a transgenic organism. There are those organisms that contain genes from a different organism. These techniques now allow for the creation of whole new organisms and spurred a huge amount of growth in biotech. You can now buy goldfish at the store that have a fluorescent green jellyfish pigment in them so that they glow in black light. It's pretty cool. They've even done this with mice, with cats. There was an artist in the New York art scene who actually, for all of his shows, because he did a lot of kind of black light art, he had created for him a genetically engineered bunny rabbit that he could go ahead and take to his art shows, and the bunny would glow a green color because it had this, uh, this gene inserted from jellyfish in its genome. Now, most of the stuff we do in terms of transgenic organisms is not something that major. It's mostly in what we call microorganisms. And if you know anybody who's a diabetic who takes insulin, they are using these transgenic microorganisms. We use them to make everything from insulin to human growth hormone to clotting factors. In the future, we may be able to use them to go ahead and produce cancer fighting agents or raw materials to make certain types of plastics. They're already doing a little bit of this with the plastics when we go ahead and start looking at the manufacture of some of our biodegradable plastics, for example. Certain synthetic fibers, we now can go ahead and have yeast manufacture spider silk, if you remember the video that we watched at the beginning of this term. So pretty cool when you start thinking about some of these applications. Most of the ones we've used today are going to be genetically engineered drugs. The first GE medicine that was produced was synthetic human insulin, because prior to that they had to use cow or pig insulin. And this was approved by the US Food and Drug Administration, the FDA, in 1982. Since then, we've had a whole bunch of other genetically engineered drugs and used them for many different aspects when we start looking at this. These drugs treat a wide variety of diseases, everything from hormone deficiencies like human growth hormone, and maybe in the future we'll start looking at things like cancer. Typically, these drugs are characterized by a very high specificity, so they're really good at targeting whatever it is they're looking for, and they tend to be safer because they're more specific. So this has been one of the big benefits of GE drugs. Genetically engineered drugs include everything from hormones to enzymes. If you have a friend who's lactose intolerant, this is what allows them to go ahead and buy Lactaid and still go ahead and eat ice cream. We've done growth and coagulation factors, antibodies, as well as many of our vaccines are produced in this way now. All of these proteins are generated using this recombinant DNA technology. So if we take a look at this, you can see here we identify the chromosome that has the trait, in this case human chromosome 11. We go ahead and remove the insulin gene and insert it into a bacterial plasmid. That gene is now part of the bacterial plasmid, and it is now a transgenic bacterial plasmid. Okay, this gets reintroduced into the bacteria. The bacteria are grown into culture. When it copies itself, because it does asexual reproduction, they're all clones. And now the bacteria produces human insulin, which can then be extracted, refined, and utilized for humans that need human insulin. And it's not just the microorganisms we've used. Transgenic plants now play a really important part of our food supply. Turns out that a lot of the plants that we consume, or wear even in terms of fiber, have been transgenically modified. Um, think about some of our super crops, right? Some of our super crops have insecticides built into them, that BT we were talking about a little bit ago. Some sort of resistance to weed killing chemicals, extra vitamins. When we go ahead and supply rice to countries that are poor around the world, we're typically using one of these super rices that has extra vitamins and minerals, okay, to help combat hunger and disease. In the future, we may find more and more plants being used to actually make materials like plastic to resist rot. We can talk about the flavor saver tomato or fight disease in other ways. And it's not just plants and microorganisms. We see this in animals as well. Transgenic organisms have been used to improve our food supply. Some of the beef that you eat, some of the milk that you drink may come from transgenic animals. Okay. In the future, these animals might provide needed all sorts of needs for humans. You see this funky picture of a mouse down here. We've regrown the structure of the tissue for a human ear. So say an ear was lost in war, and so they come back home, we can regrow an ear to surgically put back onto them. That's pretty cool that we can do things like that. Um, so there's a lot of potential in genetic engineering to produce a lot of medical benefits. But what I'm going to have you explore is, are there also potential downsides, and what are those? When we look at human genetic engineering, this shows tons of potential that we have maybe not tapped fully. The human genome has about 3 billion base pairs. And just like with any other organism in humans, this could be simply the editing of genes in living cells. Okay, you have a disease due to a problem in a gene. Let's go ahead and send in something like CRISPR to go ahead and edit that out. These are the potential futures that we're looking at. Genes, remember, are simply segments of DNA. It's just a series of nucleotides that are responsible for building proteins in the physical body, which give us our phenotype. So this includes all heritable characteristics, everything from skin color to height, to eye color, hair color, certain intelligence factors. There's so many different things that this could end up impacting. And that's beyond just the correction of disease or immunity to potential diseases. And we can talk about the fact that they've identified individuals who are immune to the HIV virus. Well, if you could genetically modify and insert that gene into other people, could we eradicate the disease? Something similar to this is happening in terms of the approved editing using CRISPR-Cas9 of individuals in England, where if they have a family history of Huntington's disease, they can go in and the state sponsors it. They will eradicate that from any, eggs or sperm to go ahead and ensure that Huntington's disease does not get passed on to future generations. Now, not all human GE is targeted at improving future generations, this kind of germline engineering. And in fact, genetic engineering today is mostly focused on what we would call gene therapy, correcting genetically defective cells in humans. This may, however, change with the advent of CRISPR-Cas9, which we'll talk a little bit about later and more about tomorrow. The first authorized attempt at human gene therapy occurred in 1990, but it was not until 1999 when the first successful treatment was recorded in bone marrow transplant patients. The process often utilizes a deactivated virus particle to insert the gene, and then still no guarantee that it's taken up. However, not all attempts are successful. Some only produce temporary relief. It depends on the type of cell, where it's located, how easily it is to quote unquote infect with these deactivated viruses. It's still considered very experimental, and there are definitely risks associated with it. Got a little video to go over it here, and then we'll finish up for today.
Gene therapy is when a gene is inserted into a cell to replace a defective or a missing gene. For example, people with cystic fibrosis are being treated with gene therapy. They're missing a gene that controls the production of mucus in the body.
How do the genes get into the cells?
That's interesting. One method researchers are trying is to attach the gene to a virus, like a cold virus, or a fat body, a liposome, and have these act as vectors to carry the DNA into the cell.
Basically, one takes advantage of the ability of the adenovirus to infect respiratory epithelial cells and uses that virus then to transport the gene which will correct the basic defect. The advantages of liposome therapy, immediately one should recognize that liposomes are not live, they don't replicate. Liposomes fuse quite nicely with cell membranes. So the idea is that one can see an easy way to target cells, albeit not particularly effective and efficient. I guess the major concerns with liposomes is that the delivery efficiency is very, very much reduced as compared to using viral vectors, on the order of 1,000-fold less efficient. What we're looking at here is a new mode of therapy, and I think society will probably take some time before it accepts it as a new mode of therapy.
So what you're looking at is two of the historic therapies that we've used in terms of gene therapy to introduce the new genetic information, either via virus or via liposome. And while we've had limited success, gene therapy is still considered largely experimental, but that may change with the advent of CRISPR-Cas9. So what exactly is CRISPR-Cas9? Well, CRISPR-Cas9 takes advantage of a naturally occurring cellular immunity system that exists in many bacteria. Okay, there's been a war going on between bacteria and viruses since the dawn of time. And one of the biggest tools that bacteria have to try and survive in this war is to go ahead and develop a CRISPR. Now, CRISPR is an acronym for clustered regularly interspaced short palindromatic repeats. In other words, you have these short segments of DNA that are interspaced with other fragments of genetic material so that it can easily identify if it gets attacked by the same virus again to try and cut it out. Okay. Now, Cas9 is another protein, fittingly standing for CRISPR-associated protein. CRISPR-associated protein 9 is a non-specific, what we call endonuclease. I want you to think of it as molecular scissors. So Cas9 creates a double-stranded break in its targeted loci. Remember, loci is position on a chromosome. And the Cas9 protein is directed to a particular target sequence by an RNA fragment. Now, if we can program the Cas9 for a particular sequence that we're trying to remove, we can actually have the Cas9 protein cut out the bad sequence and insert a good sequence in its place. And it's effective many, many times more frequently than what we saw with our liposomal therapy or with our viral vectors. And so this has a high degree of specificity, fewer side effects, and is much more reliable in making it happen. Now, tomorrow we'll go ahead and give you guys a video to look a little bit more deeply into this and what this could mean for the future. But that's what I've got you for today. I hope you guys have a great rest of your day, and we'll discuss this more in the next couple classes. All right, have a great day. Take care, guys. Bye.